Hi everyone, this is Shruti, and today I'll be talking about how we can connect metadata analysis and AI to accelerate target and drug discovery. So, before diving into our solution, I wanted to set the context. As you can see, data-driven drug R&D is a long journey. It is segregated widely into five major steps, and the solutions that I'll be discussing today currently plug mostly into the first three stages, which are target discovery, preclinical, and translational development. Next slide, please. So, with this, I wanted to just highlight that 60 to 70% of discovery time is spent on data challenges, and these challenges mostly show up in three ways. So, first, data is fragmented and inconsistent across repositories. Second, the analysis is often slow and not reproducible, mostly because it's manual. And third, we have limited actionable insights due to limited prediction capabilities. These bottlenecks add months to every discovery program. So, this leads us to a central question. Next slide, please. Yeah, so how do we move from fragmented data to confident results faster? And this is really at the heart of everything I'll be discussing today. So, we are trying to solve this problem by building an integrated system with a vision to scale data driven discovery. And we are approaching this through three key pillars. First one is metadata harmonization, which adds context to the data. Second is analysis pipelines that provide standardized workflows. And third is AI toolkits that help with efficiency and insights. Next slide, please. Yeah. So while siloed tools do exist, but the key differentiator here is integration. So it is actually bringing these three layers together that helps in accelerating the discovery process. The platform that we are building is called Strand's scRNA portal. In line with our vision, we are building an integrated workflow to bridge metadata, analytics, and AI. And I'll be walking you through each of these sections in more detail. The first pillar is metadata harmonization. This pillar is very crucial, but somehow remains the most underestimated part of the stack. So metadata adds context to the data and enables cross-cohort comparison. But the problem here, though, is that the metadata can be heterogeneous across datasets. For example, the same disease can have different labels across different datasets. I'll give you an example. So, for ulcerative colitis, which is a disease, it can be labeled as either UC or IBD or colitis in different datasets, which make comparison noisy and difficult. So, the solution to this problem is metadata harmonization, a process that involves standardizing these terms using ontologies and controlled vocabularies. So, for example, taking the same thing, right? The disease labels that we discussed earlier. So, we can map all these three terms, UC, IBD, and colitis, across different data sets into a single term, which is ulcerative colitis, using DOID ontology as an example. So the ontology, of course, here can be customized for the end users, but the end goal here is standardization. And metadata harmonization, you can imagine, thus introduces the standardization, which leads to making the data sets ready to query, compare, and build insights on. And it's really the foundation of everything that follows. And here is a metadata harmonization pipeline. So it has three main steps. The first step is data harnessing. For this, we have built an LLM powered query agent that can automatically harness relevant data sets from public databases. It also has a built in validator to QC the retrieved results. The second step is metadata harmonization. It is done manually by a team of curators by following a standardized SOP. We have over 80 metadata fields, 35 of which use ontologies or controlled vocabularies. And I also want to emphasize that our repository is very regularly updated and we add new data sets to it every month. The final step is the most crucial step, which is the quality control process. Here we have two independent teams to perform curation and two rounds of QC. which ensures very high accuracy and high quality of the datasets that we have in our repository. The bottom line here is that our process saves scientists approximately 30 to 40% of the time they would otherwise spend on dataset selection and early insight generation. So within metadata harmonization, we have first built a disease-specific repository. This is our platform. And if you navigate to the dataset section, you can see the disease areas that we have worked on. It includes IBD, Alzheimer's, Parkinson's, and frontotemporal dementia. All of these are sourced from public repositories. We have currently around 10,000 samples in our metadata repository with more than 150 curated cell types. And our differentiation here is twofold. So, first, we have disease specific datasets with broad modality coverage. So, currently, we have 9 modalities across transcriptomics and proteomics. And the second differentiator is that we have deeply curated metadata that captures granular biological context. And I'll show you an example next. So, this is where things get really powerful because through our harmonization process, we capture very granular attributes like clinical severity, disease behavior, treatment response, etc., shown on the left-hand side of the table, which allows us to define very precise cohorts. So instead of just asking IBD versus control questions, we can ask much more meaningful questions. And a live example is shown on the next slide. So this is a snapshot from our portal, which shows that we have around 1700 samples and 43 million cells from single cell experiments on IBD. And if you're a researcher who is specifically interested in understanding cell states that affect disease severity, we have a filter to capture clinical severity like Mayo score, etc. for IBD, which is shown right next to the arrow. And when we select that filter, we can shortlist samples having this information in a single click. So in this case, you see that we have 163 such samples with around 200 case cells. So this shortlisting would otherwise take days, but it is made possible in seconds through our platform. So, and once you've shortlisted the samples, you can begin with the subsequent downstream analysis to get the answers to your questions regarding the cell states affecting the disease severity. So going forward, we wanted to complement our growing repository with a chatbot agent to query the repository conversationally. So we have an inbuilt chatbot that supports multiple layers of questioning, identifies consistent patterns, and enables insight generation. The chatbot has around 90% accuracy and roughly 20 times faster than conventional coding to get the same answers. Next slide, please. As a next step forward, we also wanted to make metadata curation scalable across new disease areas. A snapshot of the tool is shown on the left-hand side. This allows users to curate around 20 metadata fields within minutes. And this automation works through a RAG LLM tool. It is also equipped with an evaluator LLM that provides quality metrics on the curated output. The tool currently operates with a 95% accuracy and saves at least 3 times the time compared to manual curation. So just to summarize our metadata harmonization pillar, we have done 4 major things here. First, we have built an AI agent to harness relevant datasets from public repositories. Second, we have a disease-specific repository with standardized metadata curation. Third, we have built a chatbot for rapid querying of the charts and tables. And fourth, we have an AI-assisted curation tool for scalable expansion. So together, all of these create a foundation layer for downstream analytics and AI. So with this, I'll move into our next pillar, which is analysis pipelines. So you can imagine once we have harmonized the metadata, the next challenge is to ensure that the data is processed consistently. And this is achievable through automated analysis pipelines. The key advantages of such pipelines are consistency, scalability, and reproducibility across datasets. I just want to highlight that even to proceed analysis, we have a data ingestion pipeline that handles metadata harmonization, self filtering, normalization, and batch correction, thereby transforming raw files into analysis ready objects. And to complement the datasets on our repository, we have built standardized analysis pipelines for both single cell as well as proteomics data. Our single cell RNA-seq analysis pipeline enables integrative analysis while preserving cellular granularity. And our post-translational modification analysis pipeline enables PTM identification, differential analysis, as well as pathway analysis. Next slide, please. So let me make the importance of metadata harmonization and analysis pipeline more concrete through a case study. Vedolizumab is a commonly used therapy in ulcerative colitis. It is an inhibitor of alpha 4 beta 7 integrin and works by reducing immune cell trafficking to the gut. The point to note here is that it is only 50% efficient. And the question is why? So, in order to understand this, we used a single cell approach. Our objective was to profile cell specific biomarkers in both blood and biopsy for the responders and non responders. To perform this analysis, we first need to get a hand at the relevant data sets. And this was made very simple through our dataset repository. We just applied the appropriate filters and we retrieved the relevant datasets and samples in a few clicks, a process again that would otherwise take days to process. So through our shortlisting, we were able to get to 3 GSC IDs from 2 independent publications, which we then took ahead to perform an integrative analysis using our single-cell RNA-seq analysis pipeline. And in doing so, we studied the cellular profiles and gene expression in both these samples and followed it by a correlation between blood and the biopsy samples. Talking about the opportunity here, it's also twofold. So, first, such kind of analysis helps uncover new targets for combination therapy. And second, it helps in identifying circulating biomarkers that can enable non invasive patient stratification. So, talking about the results of the analysis shown on the next slide, we identified 37 upregulated and 64 downregulated genes consistently altered across responders and non responders in both blood and biopsy samples. So, this such kind of analysis you can imagine are very powerful and it can be very crucial in guiding patient stratification and therapeutic decisions. I just want to emphasize that this was only made possible efficiently because we had metadata enabled cohort selection through our database and analysis pipelines that enabled integration through connected workflows. So, with this, I will summarize our analysis pipelines pillar. So, we have a a few key points to take away. We have first, we have built standardized workflows with automated pre-processing and QC that enable integration of multi-cohort datasets and can provide structured inputs for AI tools. With this, I'll move on to our third pillar, which is the AI toolkit. Within this, we have 4 different tools shown on the next slide. Yeah. So here, the 4 tools include automated cell type annotation. Second one is multimodal integration tool. Third is the gene perturbation modeling. And the fourth one is the drug response prediction tool. And these map directly to the decisions that slow down discovery programs. And I'll explain how in the next slides. So this is our automated cell type annotation tool. And what this tool does, it helps quickly annotate barcodes from single cell experiments to respective cell types. An example of the 3D UMAP generated by our tool on cells with ulcerative colitis is shown in the left hand side. And since it's an ML based approach, it also has the ability to uncover novel or rare cell types, which is otherwise not possible through marker based approaches. We also have a benchmarking shown on the top right charts. So here you see different colors for like black is for SC balance, pink is for cell typist, and green is for SCGPT, dark blue is for SCBERT, and cyan is for SCBERT GPT ensemble model, which is our model. And this the one colored in cyan, which is our ensemble model, you can see that it leads both on accuracy and macro weapon scores against the other conventional tools. So going to the next one, we have the multimodal integration tools. So today our tool is capable of combining scRNA-seq datasets with ATAC-seq datasets. And a combined embedding obtained from this tool is shown on the top right graph. So such overlaying leads to improved clustering and trajectory inference, sorry, and more holistic biological insights. And our benchmarking here again is shown on the top right charts. So our model again is represented by in the cyan color, and it's a joint SCMVP plus Surat approach. And as we can see, the clustering accuracy was across both PBMC and lymph nodes was higher as compared to the standalone approaches, which are shown in green and dark blue respectively. So going on to the next one, we have the gene perturbation tool. So this tool addresses a critical bottleneck in target validation, which is the gap between identifying a candidate gene and knowing whether perturbing it will have the desired effect. Our computational perturbation tool predicts pathway level impact for both single and two gene perturbations before any wet lab work is done. So and again here the benchmarking is shown on the left hand side. So we have again built a unified predictor also represented in cyan on the charts, and you can see that our model consistently achieved lowest mean square errors across both seen and unseen gene combinations as compared to the other approaches, which are AttentionPert, GEARS, and GEARS SC. And example of a use case is maybe you can just take an example of TIC2. So this TIC2 is an inhibitor used in ulcerative colitis treatment. And you can imagine that once you, let's say, try to inhibit the TIC2 molecule, what other downstream transcriptomic changes can follow and how this inhibition can affect the pathways. All of that can be predicted through this tool. So this is how powerful it can get. And for a discovery team, this means de-risking the target list computationally so that expensive experiments are focused on the most promising candidates. And finally, we have the drug response prediction model. So given the gene expression data and drug structural details, this tool predicts which cell lines or patient profiles are likely to respond and which will not respond to a particular drug. So here the benchmarking is shown by representing the Pearson correlation plot on the left-hand side, which shows strong alignment between observed and predicted IC50 values, which are represented in green and blue respectively. And since most of these dots, as you can see, are above the red line, it shows that our integrated predictor, which is on the y-axis, performs very well as compared to deep CDR represented on the x-axis. So such kind of tool directly supports patient stratification strategy and helps de-risk downstream clinical study design. So this basically means fewer failed trials, more targeted enrollment, and faster path to approval. With this, I'll just summarize what we have built in our AI toolkits. We have 4 ensemble models that maximize predictive performance by leveraging complementary strengths from multiple foundation models. Our toolkits enable more accurate cell type annotation. We can have better multi-omic integration. We can perform computational perturbation modeling, and we can also perform drug response prediction. And all of these applications can be plugged into different stages of the target and drug discovery process. So, to bring this talk to a close, I want to emphasize that we are building a unified workflow through our scRNA portal, where we are integrating metadata harmonization, analysis pipelines, and AI toolkits. And connecting these three layers is what turns data into faster decisions. This really has compounding value and can help in at least 10 times faster discovery, higher confidence in target selection, and around 60 to 70% gains in efficiency. As a take home message, I want you to leave with the thought that the future of the data driven discovery is not better tools, but it is in connected systems. And with this, I would like to give a huge vote of thanks to all the team members whose names are flashed on the screen. It has been a massive effort across different teams. And of course, thanks to all of you for your attention.